Assalamualaikum, welcome back to Cellumistry. Hope you are all doing good. In today's video, we are solving top 50 hard conceptual MCQs from Enzyme chapter according to the Punjab textbook board and PMDC MDCAT syllabus. These questions are designed to strengthen your concepts and prepare you for the actual MDCAT exam. Pause the video, attempt each MCQ yourself and then check the correct answer. Let's begin and see if you can score 50 by 50. Let's start with MCQ number 1. A rise in temperature from 25 degree to 35 degree centigrade increases the rate of an enzyme catalyzed reaction because Answer is C. The frequency of effective collisions increases. Which statement best distinguishes an enzyme from an ordinary catalyst? Answer is C. It is highly specific in its action. An enzyme loses its activity after being heated to 80 degrees Celsius because Answer is B. Its active site changes shape. Which group would best represent the effect of increasing substrate concentration on enzyme activity when enzyme concentration is constant? Answer is D. Curve that rises and then levels off. The lock and key hypothesis explains Answer is C. Enzyme specificity. If all enzyme molecules in a reaction are occupied by substrate molecules, the reaction is said to have reached Answer is C. Saturation point. Which factor would decrease enzyme activity without altering the enzyme's structure? Answer is B. Removing the substrate. An enzyme that works best at pH 2 is most likely found in the Answer is D. Stomach. The role of an enzyme in a chemical reaction is to Answer is C. Provide an alternative pathway with lower activation energy. A competitive inhibitor reduces enzyme activity by Answer is B. Binding to the active site. Which statement about cofactors is correct? Answer is C. They assist enzyme activity. An enzyme catalyzes only one type of reaction because Answer is B. It has specific active site. If enzyme concentration is doubled while substrate is abundant, the reaction rate will Answer is C. Approximately doubled. Which condition is most likely to denature a human enzyme? Answer is C. 95 degrees Celsius and pH 7. A student observes the increasing substrate concentration no longer increases the reaction rate. The best explanation is that Answer is B. The enzyme is saturated. An enzyme and its substrate fit together because Answer is B. Their shapes are complementary. Which statement correctly explains why enzymes are effective catalysts? Answer is B. They lower the activation energy. If the pH of an enzyme solution changes drastically, the enzyme may become inactive because Answer is B. The active site's shape changes. The maximum rate of an enzyme catalyzed reaction occurs when Answer is C. All active sites are occupied. Which factor would not affect enzyme activity? Answer is D, the color of the substrate. The part of an enzyme where the substrate binds is called. Answer is B, active site. Why can a small amount of enzyme catalyze a large amount of substrate? Answer is B. Enzymes are not consumed in the reactions. An enzyme is placed in boiling water for 10 minutes and then cooled. It remains inactive because. Answer is B. Denaturation is usually irreversible. Which statement about enzyme specificity is correct? Answer is C. An enzyme usually acts on a specific substrate. A reaction does not occur in the absence of an enzyme because. Answer is B. Activation energy may be too high. Which condition would most likely decrease the rate of an enzyme catalyzed reaction? Answer is C. Lowering temperature far below optimum. If an enzyme has an optimum temperature of 37 degrees Celsius, its activity at 25 degrees Celsius will be. Answer is C. Lower than at 37 degrees Celsius. Competitive inhibition can be reduced by. Answer is B. Increasing substrate concentration. Which statement is correct about activation energy? Answer is B. It is the energy required to start a reaction. Which graph best represents the effect of increasing enzyme concentration while substrate is abundant? Answer is C. Straight line increasing. An enzyme's active site is mainly determined by its. Answer is A. Primary and tertiary structure. Which of the following is an example of reversible enzyme inhibition? Answer is B. Competitive inhibition. At extremely low temperatures, enzymes become inactive mainly because. Answer is B. The frequency of collisions decreases. Why does enzyme activity eventually stop increasing even when more substrate is added? Answer is B. All enzyme molecules become occupied. A mutation changes the amino acid sequence of an enzyme. Which property is most likely affected? Answer is B. Shape of the active site. An enzyme has an optimum pH of 8. Which environment is most suitable for its activity? Answer is C. Small intestine. If substrate concentration is kept constant and enzyme concentration is doubled, the reaction rate will. Answer is D. Increase only if substrate is sufficient. The lock and key model suggests that. Answer is B. Substrate have a specific shape complementary to the active site. Which statement is true regarding enzymes? Answer is C. The lower activation energy. A student increases temperature from 20 degrees Celsius to 40 degrees Celsius and observes an increase in reaction rate. The best explanation is. Answer is B. Effective collisions increased. Which statement correctly explains enzyme saturation? Answer is B. All enzyme molecules are occupied by substrates. An enzyme catalyzed reaction stopped because all substrates have been converted into products. Which factor is limiting? Answer is C. Substrate concentration. Which factor is least likely to affect enzyme activity directly? Answer is D. The color of the reaction vessel. Which of the following can permanently destroy an enzyme's catalytic activity? Answer is D. Increased substrate concentration. The active site of an enzyme is formed by. Answer is B. Specific folding of the protein molecule. Why do enzymes speed up reactions without being consumed? Answer is B. They are regenerated after each reaction cycle. A mutation changes the tertiary structure of an enzyme but not its primary structure. Which property is most likely affected? Answer is C. Shape of active site. Which statement about competitive inhibition is correct? Answer is C. The inhibitor competes with the substrate for the active site. Which pair of factors can alter the shape of an enzyme's active site? Answer is A. pH and temperature. A student adds more and more substrate to an enzyme solution. Eventually the reaction rate stops increasing because. Answer is C. The enzymes are saturated and working at maximum rate. Comment your score out of 50 below and let me know which biology chapter you want next. If you found this video helpful, don't forget to like, share and subscribe to Cellumistry. Here you will get chapter-wise MCQ, PMDC syllabus guide, study plans and complete MDCAT 2026 preparation. I will make next video on acellular life MCQ. Thanks for watching. Allah Hafiz and keep learning with Cellumistry.